Hello guys, I am Shubham from ShubhVed Tutorials. This video is about human karyotype. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. From this video, we are starting with the cytogenetics part, and in that we study human karyotype. Basically, first of all, what is cytogenetics? Cytogenetics is the word combined with two branches, okay, that is cytology and genetics. Both these branches are combined, okay. In this we study the chromosome, all about chromosome, okay, all about chromosome, all about chromosomes. It may be structure of chromosomes, it may be function of chromosomes, or it may be abnormalities of chromosomes, right? So we know that chromosome is made up of DNA and proteins, right? So we study structure, function and abnormalities of chromosome in this cytogenetics, and human karyotype is one of the method, okay. In human karyotype, now human karyotype is what? This is a, we can say this is a, in this we discuss about chromosome structures, okay, structures, then shapes, size of chromosome, okay, then the numbers, these things we study in the human karyotype. It is known as karyotype analysis also, we can say. This is used in medical genetics to understand the different chromosomal diseases, okay. So characteristics of human karyotype. First point is about chromosome size, right? Then chromosome number, sex chromosomes, centromere position, then nucleolar organization, nucleolar organizing means how nucleolus is organized inside a nucleus, right? Means all we discuss here about chromosomes. Then heterochromatin region, then we can say secondary constriction also, and banding pattern. Banding pattern means staining pattern just of chromosomes, right? So these are the useful characteristics of human karyotype. So first of all, chromosomal classification. Chromosomes are classified according to position of centromere, okay. It is known as Denver classification. Denver classification of chromosomes, which is based on position of centromere. Position of centromere. So we know that this is a chromosome. And see, this part, we can see a centromere here in a blue color, right? Yes, centromere. When it is located in the center, it is known as metacentric. Okay, it is known as metacentric. When centromere is in the center, when it is little down from the center, okay, little down or up, it can be little up from the center or down the center. So it is known as submetacentric. Submetacentric. Okay. And when the chromosome is near the top, near the top, near the top, it is known as acrocentric. Acrocentric. And when it is located fully at the top of the chromosome, it is known as telocentric. Telocentric. So these are the important terms of centromere position and the chromosome. So metacentric chromosome, submetacentric chromosomes, acrocentric chromosomes, and telocentric chromosomes. Okay. Now the next thing about chromosome karyotype is this. This is the karyotype, known as karyotype, or we can say karyogram, when it is plotted on the paper, right? When it is plotted on the paper. Also, it is known as idiogram. Okay, idiogram. In this, we can see the chromosome structure, right? So, this we have already discussed that in humans there are 23 pairs of chromosomes, total 46 chromosomes. One pair we get from mother, one pair we get from father. Same chromosomes are known as homologous chromosomes, and different are known as non-homologous chromosomes. So, all these terms are important ones. Now, from this, in male, sex chromosomes are X and Y. Female sex chromosome 2X, right? So, how we will present it? For males, 46XY, males. And here, in females, 46XX, right? Then, from this, now we will discuss which are the submetacentric, which are the metacentric, which are acrocentric, which are telocentric, according to the numbering. So, according to Danvers system of classification, see 22 pairs of human chromosomes are placed in 7 groups. Okay, so first group A, position of centromeres here given in the second, then size and idiogram number. As we saw the idiogram, this is a idiogram, or we can say karyogram. Okay, these are used interchangeably. So first group contains large size of chromosome, metacentric. Okay, 1, 2, and 3. In second group B, submetacentric, size large, 4 and 5. Okay, then third group C, submetacentric, medium size of chromosomes, and the chromosome numbers are 6, 7, 8, 9, 10, 11, 12, and X. Fourth group D, acrocentric chromosomes, medium size of chromosome, and 13, 14, 15. Again, the next group is E, submetacentric. Submetacentric, slightly small size. Okay, slightly small size, 16, 17, 18. Then F group, metacentric chromosomes, small size chromosomes, ideogram number 19 and 20. G, the next group, contains acrocentric chromosomes with small size of chromosomes, and 21, 22, and Y are the chromosome numbers. So we can see that the chromosomes are arranged according to the sizes as they are decreasing. Starting in they are large, right? 1, 2, 3 large, then medium, then small. Okay, X is also large, it comes in medium size, right? And Y comes in very small size, and it is a acrocentric Y, right? So this is we have to remember. And also the next concept is the symbols we have to understand. Okay, symbols in karyotype study, that is a long arm and a short arm, right? Short arm is known as P, long arm is known as Q. Okay, then if I write 2p means short arm of chromosome number 2. If I write 5q means long arm of chromosome 5. Okay, as well as there are some chromosomal abnormalities like del means deletion, inversion, then duplication. Okay, translocation. Translocation can be written as 2 and 5, means there is a translocation, reciprocal translocation between chromosome 2 and 5. Okay, and if deletion, we can write deletion 2 means deletion in chromosome 2, inversion in chromosome 5, if we want to specify the chromosome number, right? Then as well as duplication in the chromosome 6, we say, right? So these are basically studied in the diseases according to the phenotype we see, right? Then plus or minus means gain or loss of chromosome, plus or minus. CEN for centromere, TEL for telomere, 2p minus means what? There is deletion, minus means deletion of chromosome, short arm of chromosome number 2. Okay, suppose I will write here deletion in the short arm of chromosome number 2. Okay, so these are the symbols we have to remember. So that's it all about human karyotype analysis. If you find this video helpful, do let us know in the comment section. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever new video is out. Also follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.